A warm greetings to everyone. We are Nano Era. Our project title is Nanomedicine in Drug Delivery. We are from Avila Convent Matriculation Higher Secondary School.
Major problem in delivering drug for a particular disease is that they damage the healthy tissues and most of the medicines is lost before they reach the required target site. For example, in cancer treatment destroys both the healthy and the cancerous cells, thus weakening the patient. We have brought in nanomedicine as a solution for this. It is a new but yet rapidly developing branch of science in which nanoscaled materials are used as carriers for the medicines. The most important carriers we use are cold nanoparticles, chitin, liposome, xanthan gum, etc. Of liposomes, they have a bilipid layer so that when we send the nanomedicine into our body, there is less chance of the medicine to be dissolved in our body fluids. Usage sector. It's widely used in treating cancer, HIV, diabetes, etc. And many pharmaceutical companies have undertaken this method of treating cancer. Benefits to the customer. They have low side effects and faster recovery, an efficient way of giving medicine, and they are the third generation in the medicines field.
In order to cure the brain tumor, the drug must pass through the 5 barriers. The first barrier is the stomach. Due to the acidic environment, the drug is degraded. The second barrier is the intestine wall barrier. After 37 minutes of struggle, the drug enters the intestine and due to the absorption process, the drug gets degraded. The next is the liver, the organ which is responsible for reducing the harmful effects of the medication. So the enzyme in here gets degraded. Protein and enzyme in the bloodstream is the next barrier. The protein and enzyme bind to the drug, which in turn results in loss of medicine. The last barrier will be the blood-brain barrier. This is a protective barrier which only allows fat-soluble molecules to pass through it. This is a total loss for the drug. Therefore, 95% of the medicine is wasted.
How does nanoparticles work? They work on many factors. The first factor what we have taken is size and geometry. The normal cells will be in a tight junction so that nanoparticles cannot pass through them. But the tumor cells will have junctions which will range from 200 to 1200 nanometers in size so that a very small nanoparticle can pass through and land in the tumor area. Now, these nanoparticles have to be activated in order to give the medicine. The nanoparticles, for example, the gold particle is sensitive to light. So, similarly, the required sensitiveness is given to the nanoparticles so that the nano shell is broken and the medicine is deposited at the required site of tumor so that the tumor is targeted efficiently and destroyed fastly when compared to other methods of treating cancer. The other method is anti-affinity ligands. In affinity ligands, they are the particles, the nanoparticles, which are very much attracted towards a particular area. For example, cancer cells' DNA are very much attracted towards gold particles, and that's why they use it. Such techniques are used, and this is how it works. And there are many other ways in which they also work. This is our prototype model. We thank everyone for giving us this opportunity to take part in this grand event. We hope that our video will be a solution to this problem. Thank you.